So good afternoon, everyone. Happy New Year. Welcome to the fourth edition of the Publique Research Club. Thank you for joining us today. This series is organized by the INSERM Publique Health Institute and IRES with the aim of showcasing innovative research, fostering scientific exchange and building stronger connection across discipline and institution in public health. Today, we are delighted to focus on the topic genomics, imaging, and AI, three technologies which are transforming research and healthcare. I let our director, Robert Barouki, introduce our speaker, Iwan Birne.
Yes, thank you very much for joining today, and thanks for Iwan to accepting to giving us this talk. Actually, Iwan is at EBI and he's involved in many different areas at EBI. And I met him a few months ago in Brno. We had a very nice meeting. Different people from actually all over the world met to discuss the possible interaction between exposome and genome. And following that, I thought that it would be great to hear Iwan giving us his talk. What I remember very well, Iwan, and thanks you very much for being here, is scale. I mean, we talked about cohorts, and you kept mentioning scale. So that's it kept it in my mind, and I'm really looking forward to hearing you today. And without too much introduction, sorry, and we will have I think more discussion afterwards after you give your talk. So please, if you want to share your slides, it would be great.
I will stop mine. And just so you know, the presentation will last about 40 minutes and will be followed by a Q&A session of 20 minutes. So please feel free to share your question in the chat. So, Iwan, I let you the floor.
Yeah, does that work? Can you see my slides?
Yes,
Yep.
You can see it.
Right, so thank you very much for the invitation. It's wonderful. There's a part of me that would prefer to be in Paris, but I would, this is the next best thing, perhaps, for this. Just some context. I'm going to do two things. I'm going to give you some context about EMBL, genomics, imaging, and AI, and then I'm going to dive into a very specific method that my research group has been involved in, which has a strong focus on epidemiology, basically. So many of you will know EMBL. We're the European Molecular Biology Laboratory. We are an international treaty organization like CERN. CERN does high energy physics. We do DNA, RNA, proteins. We're headquartered in beautiful Heidelberg, Germany, and we operate from 6 sites across Europe. 2 at some of the world's best synchrotrons for X-rays in Grenoble, in France, and in Hamburg. Then there's EMBL EBI, which is just south of Cambridge in the UK at Hingston. That's where I'm physically based, where I am today. And we do bioinformatics, data, basically, and analysis. And then there are 2 other sites in Rome, which focuses on epigenetics and neurobiology, and in Barcelona, right by the beach, actually, focusing on tissue biology. We're owned by 29 different countries, basically Europe plus Australia. We have 5 different missions to perform excellent research, but also to deliver scientific services. And from this perspective, a really important one is data. So we host the world's most comprehensive set of biomolecular data, and that includes ENA, AlphaFold DB, UniProt, Ensemble, Europe PubMed Central, and many others. We perform advanced training, both the classic postdocs and pre-docs, doctoral students, but also things like specific engineering training courses and training secondments. We work with industry across Europe, and we integrate the life sciences both across Europe and globally. I want to now give you a very big picture, strategic view of AI. And one aspect, and it's great, Robert, bringing up Breno and the wonderful Exposomics meeting we're there, because that was actually where this gentleman, of course, Gregor Mendel, was from, the monk from Moravia, now the Czechia. And Mendel was arguably the first data-driven biologist. So Mendel and Darwin, very similar time. They were born at a very similar time, and they did their science at a very similar time, but they did it in a very different way. Darwin took many, many observations himself and took letters from many people, but wrote very long books. They're just books which you read, and it's a piece of logic. But Mendel did something different. He designed experiments, and then he tabulated data. He generated data, and from that data, he derived two different things. One thing was an understanding of the world, in this case, genetics. And secondly, from that understanding, he could make accurate predictions. And there's an argument that we are all following in the footsteps of Mendel. We generate data now, not just by observations using our own eyes, but many other ways. We lay that data out, and then we derive things from it, both understanding and prediction. And of course, this has become not just sort of simple statistics, it's become increasingly sophisticated, of which the latest one is AI. So I want to talk first about the two driving data generation technologies in molecular biology. The first is about genomic sequencing. I'm just showing here the different technologies that have been involved in genomic sequencing over the years, ending on the right-hand side with Oxford Nanopore and PacBio, these single molecule reading schemes. And just to mention, many of the attributes about how we measure molecules have gone up. Firstly, the number of simultaneous molecules have gone from one at a time through to billions at the same time. The length of the DNA polymer that we can read has gone from only 20 odd bases up to a megabase or beyond a megabase. We can read not only our DNA, but also RNA and modifications like epigenetic modifications like 5-methylcytosine. A big driver for research has been the cost per base pair measured has gone down by orders of magnitude, many, many orders of magnitude, so over 1 millionfold. The error rate has also improved. In fact, it was reasonable. Oxford Nanopore at one point was 1 in 10. That's okay if you're a computer scientist, but it's a bit scary if you're not. But actually, all things, including Oxford Nanopore, has improved. And the error rates are increasingly low. And then another important aspect for clinical colleagues is the all-in time to result has really reduced. And the most striking example of that are people in Nottingham and in the Netherlands and increasingly in America, where they're doing intraoperative DNA sequencing of tumors from people with brain tumors. So during the operation, the tumor is extracted, removed, tumor tissue is removed, it is sequenced, and enough is generated in under 2 hours to allow the clinician to make decisions about the ongoing operation. Now, a really important thing here is there's no inherent biophysical trade-off between these axes. And in fact, there's going to be a future of much more development, I think, in all of these axes around genomics. And this has enabled many things, and I don't have time in this talk to go through them. You'll be delighted to hear. But of course, there's reference genomes. I was involved in the original human genome reference. It's printed out as a book in the Science Museum, which is rather wonderful, in London. But we are now sequencing the reference genome of many, many species across the planet. Something which is a big part of research is the fact that if you can make your experiment become a DNA sequencing experiment, if you can get a readout via DNA, you can make it scale. And that's what we call things like RNA-seq and ChIP-seq and everything else, --seq. The scaling of DNA is really remarkable. And then I know that France has made great inroads into this, as has many other countries, which is the integration into clinical practice. It's really transformed rare disease. It's increasingly transformed cancer. And it's also, and COVID really showed this, our ability to transform our understanding of infectious disease, including fast-moving epidemiological outbreaks. So I want to touch on the other key technology here, which is imaging. We can now span many, many different scales by imaging using different modalities. And this goes from the molecular atomic scale using X-rays and electrons through to entire tissues, which of course involve things like MRI, echo-based systems, and optical systems. And an important thing here, by the way, is that we can do this at many different scales and time resolutions, and we really have a comprehensive spanning of all scales and fields of view now. I just want to touch on some of the impressive improvements we have. This is mainly from work that happens at EMBL. The left-hand side is from Julia Mohammed's lab, where there is looking at ribosomes in intact bacteria, frozen, cryo-frozen, and seeing the action of antibiotics in the cells, in the bacteria themselves. So seeing the structural changes of ribosomes in bacterial cells due to antibiotics. The middle one is work from Cornelius Gross and colleagues in ESRF in Grenoble, looking at making very, very rich and detailed views of tissues, in this case the mouse cortex, allowing you to resolve synapses in a very wide field of view. And the right-hand side, I won't go into it, it's a new type of microscopy looking at mechanical properties of cells using something called Brilleen microscopy. Now, I could have talked about those technologies more and more, but I really want to move on to this, which is how do we do the derivation of understanding and how do we make good predictions from these large data sets? And this is where AI and machine learning has really changed what we can do. And I think of there being two major engineering drives to AI. One is the accumulation of lots of very good data. And just to remind you, EBI is that source for the world. Although we're based here in the UK, we're owned by 29 countries, including France. Imagine a little tricolour above us. This is a little bit of France in Cambridgeshire, UK. The other combination is effectively the ability to do very, very large scale differentiation. So as many of the people probably on the call know, if you do machine learning and you do AI, you do gradient-based optimization, and that hinges on the fact that you can make a computational differentiation of your function. And so you have a very, very large function, maybe with 1 million variables, maybe with 1 billion variables. You computationally create the derivative of that function, and that allows you to optimize these crazy big models. To do that, you then need a piece of hardware. Those hardwares, at the moment need GPUs to work well, though other solutions are available. And then you can do many different things. And I'm going to touch on the first two lines here, labeling and modeling. And then what we're really going to dive into is modeling-based deep learning. So labeling is a really important part of biology where we have a data set and we know there's information in there, but we want to surface or bring out that information and make it a very concrete aspect. So the left-hand side here is taking an MRI image of a heart and labeling the different chambers of the heart. Previously, a human individual would have to click on each voxel. These days, this is very reliably done by AI-based methods. Similarly here, the middle one here is actually a plant-based cell. Very similar problem. You want to label where all the cells are. Again, humans previously had to do that, but now AI can do this extremely well. And actually, just to show that this is not just about images, Here's a problem in oxide nanopore, which is that you have an ionic current. Those are the dots at the top of the screen there, and you need to change that into a base pair view prediction that is kind of deriving a piece of information from that signal. And again, we use AI for that. But then we have another kind of class of problem where it's less about labeling a data set and more about making predictions. This data is equivalent to this, or this data, something is going to happen in the future. I could spend a lot of time talking about AlphaFold, the AlphaFold database, which we were a big part of, both in helping Google DeepMind access the data and in allowing a way for Google DeepMind to publish all the AlphaFold results to the entire world. There are ways of interpreting the genomes, things like Informer, Chrome BPNet, from my colleagues in Stanford, and also colleagues in Google DeepMind. But what I'm actually going to talk about is the right-hand stuff, which is really bringing AI to epidemiology. Actually, at the start here, I'm not going to use imaging and I'm not going to use genomics, but later on, I'll show you how we bring that in to model, in this case, the future trajectory of people's healthcare. So what's going to happen in the future for individuals? So this is a piece of my own research. I've done this in close collaboration with Maritz Gersten from DKFZ. This was the Nature paper published on this. And we made an observation that healthcare is very similar to language. And we have a very powerful technique for language called generative pre-trained transformers. It views languages as a series of tokens. This is a classic English sentence. The quick brown fox jumps over the lazy. And the goal is to predict the next sentence. And dog would be the next word, in the sentence. Now, amazingly, you can not only have these predict one word, but the context of these processes allow you to predict many, many words, so much so that it feels like these things are talking back to you. And these are OpenAI, the chatbots, the different AI techniques that you use these generative pre-trained transformers, and they can capture a huge amount of context and a huge amount of information from corpuses around the world. So at some level, healthcare is a very similar problem. We can think of healthcare events as tokens. Here are a number of tokens that happened in someone's life. And wouldn't it be great to be able to predict what will be the next token in someone's healthcare trajectory? This would be a very useful thing to do. But there is a very different, one big difference between healthcare and language, which is that there is a variable amount of time between the tokens. And that is, we don't have that aspect of variable time in language or variable gaps in language. And if you just take the generative pre-trained transformers as they're used in language modeling and you apply them to healthcare, it doesn't work very well. I mean, it works as in the code runs, but it does not produce accurate predictions. So we adapted the generative pre-trained transformer in a variety of ways. The most important thing was enforcing a prediction of time to event. And so we have an explicit model of time. It's a series of exponential decays. And our model is forced to predict the time as well as what the next event is. And in fact, in many ways, we weight the time prediction more than the next event. The next event is a very simple derivation of the time prediction. And there's some other things which maybe in questions you might want to ask about how we handled, which are basically all about different aspects of time in this. Just to mention the data sets that we used, and Robert was mentioning scale. Scale is really quite important here. So we train inside of the UK Biobank. We actually don't train across all of the UK Biobank. We held 20% out as a zero-shot test set, validation set. We also hold a number of years out as a zero-shot test set. But perhaps our most challenging data set was predicting inside of Denmark. I must stress, we never trained our model in Denmark. So we only tested our model in Denmark. And so when we come to the Danish data, that is our strongest test of the generalization of this. So in this prediction scheme, we can predict, or the model would like to consume all the tokens. We're using ICD-10 codes as our tokens, and we use the tokens where we have enough data. That's 1,261 different diseases. I'm just showing you here 8 of those diseases, including the rather ominous death token, by the way. Which you have to add. It's not an ICD-10 token, so it doesn't have an ICD-10 number here. So what you're seeing in each of these plots is age is on the x-axis, and then on the y-axis is a logarithmic scale of the rate of this particular disease happening. And the very light points, which you can see in the background, those are a subsample of all the points across all of our validation data set for everyone in there. And you can see they have varying rates. The rates go up and down, as it were. Different people have different rates, and often those rates change. The average changes over time. The solid lines are the population averages of the rates. So if you just very crudely take all the men and all the women and you say, what is the rate of, for example, breast cancer, that's C50 in the top right-hand side. I hope you can see my cursor. You can see that this solid line here is that average rate across all women, and this is the average rate across all men. And men do get breast cancer, but at a much, much, much lower rate. And you can see that this cloud of points, the solid line is through the cloud of points, the light points. The dark points are the token just before this individual got a diagnosis of this disease. What's important here is that more of the dark points are above the line than below the line. In other words, we are making a rate prediction for this individual that they are very likely to get breast cancer in the next year. And it's way above the age-sex, the crude age-sex control. So this point being up here. Now, if I go to death, you can see that we have many points above the line here. If I go to septicemia again, or depressive episode. So the fact that we have many of our solid points above these lines indicate that we are predicting for the people who do go on to having that disease, we are predicting a much higher rate of that disease occurring for that individual. Now, there are many ways of summarizing this statistic. We actually, it's quite a tricky thing to summarize, or there are many options about how you summarize it. And I'm just showing now for 3 diseases, one way of summarizing our performance, which is area under the curve, AUC curves. Our model is called Delphi, it's in black, and you see a number of other models, which are in the other colors here. Some of these models, for example, the Charlson score for death, or the UKB DRS score for dementia, or the QRIS score, are used in the clinic now. These are used in the clinic in different countries now. And what you can see here across these 3 diseases is that Delphi is the top, or very, very close to the top predictor. But actually, say for dementia, it's very similar to the best model used in this case in the UKB DRS. But importantly, Delphi can do this across all diseases. We can do this not just for one disease, we can do it for all diseases simultaneously. And that gives us some power and some possibilities that these individual models do not have. And then you notice for death, we are actually, significantly better than everybody else. Now, I showed you there are 3 diseases. This is now getting to all 1,261 diseases. The top panel here shows the area under the curve, and 0.5, as people know, I'm sure, is random. 0.5 is a random prediction. So you can see most of these points, this is in the longitudinal validation, most of these points are above 0.5. There are 1 or 2 which is dipping below, and that is a kind of sample size problem, we believe. But actually, this AUC metric is a bit misleading because the big driver for most diseases is age and sex. And if having a good AUC for disease prediction can be trivial for some diseases once you consider age and sex. And so we focus more on this bottom line here, which is the difference in AUC versus an age-sex control. And importantly here, we're mainly positive. We're above 0. 0 now would be the same as an age-sex control, and we're mainly positive. And in some cases, we're significantly better than an age-sex control. Now, this is looking very much at the next token, or 3 months out, or what? Oh, so sorry, before I do that, let me go to Denmark. This is exactly the same model. We do not change the model. We just took the model that we trained in the UK and we apply it in Denmark. And it's actually more people, so 2 million people. So this is a very large sample size and a very big test of the model. And you can see now that we do get better performance or good performance. AUCs above 0.5 and above age-sex control. And just to emphasize, we have never seen any Danish data. A question, you'll notice that there's one set of diseases which we're not so good at, which is cancer, where in fact we don't do here in Denmark, as well as an age-sex aspect. This, we believe, is very much a very complicated technical issue, and we don't believe that's anything to do with the model. And you can ask me questions if you like about that. That's quite a long story, so I will not bore you with it at the moment. But we think it's solely technical, solely ascertainment. Now, another important thing, AUCs tell you that you can rank patients appropriately, that the top ranked individual is the most likely to get the disease. But it doesn't tell you whether you've scaled that ranking correctly. And this is looking now at an important question about calibration. Our rate predicted by our model is on the x-axis here in a logarithmic scale. And the observed rate from the validation data sets are present on the y-axis. It's a bit jumpy because we don't have enough numbers. This again goes to the need for really quite large scale for you to be able to make all these estimates. But the important thing here is across many, many diseases and across different sexes and across different ages, our rates are well calibrated. In other words, these lines lie around the x equals y line, roughly speaking. And so we can really trust that our rates seem to be they seem to be good predictions of the actual rates that that occur. And this allows us to do something which I don't believe we've ever been able to do previously in epidemiology, but we routinely do in chatbots and AI systems, which is we can sample future trajectories. So when you write a message in ChatGPT or something like that, it doesn't just produce one word, it produces many words. And what it's doing there is it's sampling from its model what it should produce. And we can set this problem in the same way. We can kind of stop the point at a particular point in time. Or of course, we can take people now, or their health trajectories up to now, and then we can simulate their future trajectories. And that's a probabilistic simulation. We can actually sort of do 100 simulations or 1,000 simulations for each individual. Now, this plot in B shows that our simulated rates, so when we look at those simulated rates, they match to observation. When we go back in time, we stop the clock in 2015, we simulate a decade's worth of data, and then we compare that to what was the actual outcome. And then the right-hand side here shows that across many, many different time windows, perhaps unsurprisingly, our performance, the simulation is better. And again, we set ourselves not against a kind of, random model. I mean, what would be the point of a random model? But we think the right baseline here is an age-sex control, and that is shown in blue. And our yellow is the proportion of correctly predicted tokens from our model, Delphi. And you can see it's better in a very short time window, but it actually persists at a reasonable rate out for 10 years. Now you might, I don't know how much familiarity people on this video call has about AI techniques. AI techniques can feel very black boxy. Actually, in my experience, they are not. They are very, you can really look at what the AI system is doing and how it's thinking. And I'm going to now show you the kind of underlying kind of how AI thinks about this. So firstly, there's an internal space of that we use. In our case, this is one of the hyperparameters we had to discover. We use a 120-dimensional space. And when we train it up to make accurate predictions, this is a projection of the 1,261 diseases into that 120-dimensional space. Now, of course, I can't get 120 dimensions easily on a 2-dimensional screen. So you then take the 120-dimensional space and you project that onto 2 dimensions using this technique called UMAP. It kind of works. It also doesn't work. So please don't overinterpret. Distance in this plot in particular doesn't really mean anything. But when things are close by, that is meaningful. So things which are close by are meaningful. And we've just highlighted in this space a number of things which are close by. Let's go to that top left-hand side first. These are the ICD-10 codes. You can see it, E10, E16, those are diabetes or pancreatic ones, but then also some comorbidities with diabetes, like retinal disorders and polyneuropathy. In fact, if we went down an ICD-10 code, these would be the diabetic examples of those ICD-10 codes. Again, going down to the bottom, neurodegeneration, we get a little cluster here around neurodegeneration, which went to the same space. The top right is putting breast cancer, but ovarian cancer also together, which makes sense. And then the bottom left, that black dot is the rather ominous death token that you don't want to get in your healthcare stream. It shows some of the other key conditions close to that, which, for example, includes acute myocardial infarction, no surprise, but also things like septicemia, which also, again, make a huge amount of sense. Now, that was one view around diseases. We can also kind of project how our AI model works using something called SHAP analysis. This is a commonly used technique in AI to try and kind of marginalize the predictions or create a view of what is driving particular parts of the AI prediction. The x-axis here is what is being predicted. So predicted token, notice the black column is the death token. And the y-axis here are tokens earlier on in an individual's healthcare stream. And red means this token was very informative about the prediction to the x-axis. And blue means it was negative. It was risk reducing. So red means it's risk increasing. Blue means it's risk reducing. Just be on the sort of on the left-hand plot here, over here, you can see neoplasms, that's cancers. They really associate with their risk increasing of death. No surprise here. You can see some other things, nervous system diseases, there's some with death here. Now, no surprise here, the pregnancy and childbirth tokens very much associate with the pregnancy and childbirth tokens. So you can see this block here makes complete sense. Let's look at another one, mental disorders. These are what we might call comorbidities, especially in later life. But pregnancy, there's obviously a whole series of tokens which happen in a very structured way around pregnancy. Now, this was very short-term view. If we now go to the long-term view, some things do persist over the long term. So, for example, mental disorders, even tokens from 10 years ago are very informative about whether your next token will be a mental disorder token, or your token in, 10 years' time. There are other things, but notice, for example, that pregnancy square has now really gone away, which again makes sense. It doesn't, whether you were pregnant 10 years ago really doesn't predict whether you're going to be pregnant now. So that makes complete sense. And it gives you some sense of the kind of querying of the system we can make. So everything I've been doing at the moment is classic epidemiology, ICD-10 tokens only. I've not used any other measurement in the system. But we can put pretty much anything that AI can ingest can be put in. So for example, we can put in environmental measures such as particulate matter or PFAS or PFOS or exposure aspect when it's been measured. We can do drug prescriptions. Drug prescriptions are very interesting, actually, because they change trajectories, but they're also predictive of healthcare events because clinicians prescribe drugs for good reasons. So they have a really interesting behavior in epidemiology, actually. And we can put in, just as we can fuse video and text in chatbots, we can fuse images or metabolomics and healthcare tokens. So cardiac MRI scans, OCT scans, retinal scans, full blood metabolome. And finally, we can put in genetics. We put the genetics token in just after birth as a way of doing this. So I just want to show you one piece of genetics we're doing. This is looking at adding in the MHC, probably the most important genetic locus here. And we're looking at a valid test set, so zero-shot test. The AUCs, this is across all disease tokens without the MHC and then with the MHC on the y-axis. And you can see pretty much all the points are above the x equals y lines. And the ones with the biggest deviation, I haven't highlighted them, are indeed things like autoimmune diseases and diseases that we know are associated with the MHC. So with that, thank you very much. I hope that was interesting. I'm very, very happy to take questions.
So thank you very much. This is really interesting. It's really, really interesting going from really very molecular aspects to some of the tools to this kind of prediction in human diseases is really very exciting. I can start with some questions. If you, instead of the Delphi, used, for example, polygenic risk score with age and sex, what would that do compared to the Delphi? Would that be similar or better, less?
I mean, Delphi is definitely better than a straight polygenic risk score, but of course you're allowed to put all of them together, and that's kind of what we're doing with the polygenic risk score. In our paper, we kind of crudely blend the polygenic risk score with Delphi, and it does improve things. Not massively. What we're finding, unsurprisingly, is that when we integrate genetics in, it really improves the prediction for early onset.
Okay.
Yeah, and not for late onset diseases. Even for the same disease, it improves earlier onset prediction than later onset prediction, which is entirely what you would expect. As you accumulate more health events, the health events are better predictors of your internal state than your genome.
Okay, but for long-term future state, the polygenic risk score does not add much. Is that what you're saying?
Not long. I mean, it's more that early onset, early onset diseases, genetics looks the most powerful for earlier onset. Okay. Which we've known, in some sense we've always known this, but it really makes it very, by integrating an epidemiological view and a genetic view into the same with a time dependence, you can see how that's used.
Okay. And you mentioned air pollution at one point. I mean, at least the PM. Does that also add much, or?
Yeah, we're only at the start of processing that. We are just assembling the data sets at the moment, so we haven't done that test
yet.
Done.
Okay, this is currently done. Okay. A question from the audience?
There is a question from the audience. So would a model work for rare diseases as the onset is quite early.
Yes, I mean, we haven't done much on rare diseases. I mean, one of the problems is that they're rare, and our model stabilizes when there's about 500 to 1,000 points incidences in the data set. And to assess it, you probably need at least 200, 300 points in there, the validation data set. Now, that makes rare diseases challenging. We will need European-wide data sets to do that. We're working with colleagues in Finland, Denmark, Norway. I'd love to work with colleagues in France on this precisely to get at that sort of thing. It is complicated. You need a lot of harmonization first.
But we can put you in contact with people because I mean Finland is a small country. I mean I know they have excellent registries, but I mean how many rare diseases would you get? But maybe countries like Germany, France, and Italy would provide much more.
Yeah. I mean this is why the UK, which has excellent also for rare diseases, you really want the genomics, so you need good genomics as well. So Finland, it is a small country, but it has excellent genomics, and it has really good haplotypes. So Finland and Denmark are quite interesting, both for their data and for their genomics. But yeah, bigger countries like France, Germany, and Britain, UK are places where we have more scale, and that those will drive. The scale is, as I said, Robert, scale is an incredibly important driver for this.
Again, great. Okay, another question.
Yes, someone says, what about C4P? I don't know. I think it's related to Finland. Or maybe.
No.
Maybe you can precise your question. We have another one. Meanwhile, can the model predict if particular chain of multiple events, disease, will cause a particular disease?
Yeah. So there's one very clear-cut area where this happens, this chaining happens, but it's less what you might call predictive of disease and more about modeling the diagnostic process. So let me give you a classic, which is that if a patient comes in with kind of crazy glucose, they usually get this very generic term first, both in the UK and Denmark. They get a very generic ICD-10 code first. And then that will usually resolve to a type 1 or type 2 diabetes code. Sometimes it resolves to, hemochromatosis. But that's very rare. So, really weird glucose, you get one code, 2 or 3 months later, you get a second code. Some of these diagnostic chains are even longer than a kind of 2-code chain. And Delphi is very good at modeling that. Of course, it's not really predictive. It is formally predictive. If you think about it from the AI perspective, this is making a prediction, but it's not risk prediction in an epidemiological perspective. Now, I think of this very much analogous to grammar. So like in language, these large language models learn the grammar of the language. Without any formalization, you just give it a lot of data, it will learn the grammar. So it'll produce grammatically good English and grammatically good French and stuff like that. So Delphi will produce grammatically good healthcare, from using that analogy for this. But we have hints at, but we need, again, bigger data sets to do the combinatorics. We've looked at whether kind of complex comorbidities and orderings of the comorbidities are important. I don't think we have the data set size to remind you, everything is going to work to the square or to the cube of the frequency of your things. So you have a n squared, n cubed, n to the fourth problem as you start to try and think about that problem. So we are going to need bigger data sets than 500,000 or 1 million.
So let me just try to get it a little bit straight. Do you mean that in the sequence of diseases that you are using to predict the order of the diseases has an influence? For example, if you get asthma before getting diabetes or the reverse?
Yeah. So we think that's true. It's obviously true for this diagnostic prediction, right? There's certain codes that must happen before other codes, and Delphi nails those. Absolutely gets them perfectly right. But that's not the question. That although that is a good prediction of of orderings, and ordering is incredibly important. That's not the question that you're asking, and we need bigger data sets for that.
Okay.
I have a few more questions. Someone is asking, do you think the model would have the same result if it was trained on the Danish data and tested on the UK data instead?
Yeah, I mean, we've done that. My colleague Andrea Ganna in Finland has done that in the most comprehensive way. And he has trained he's taken the UK trained data and tested on Finland. He's taken the Finnish data and tested on the UK. And you know, the model does best in its own data set. But the cross, it works pretty well in all directions. We think if we want to go down an ICD code, we're at ICD code level 3. We want to go down to 4. I think there will be more system level questions or complications when we go down to ICD level 4. So I don't want to say that this generalization will work at all granularities. I'm not sure that will be the case.
Thank you. And the person added, how does the model handle imbalanced data?
Yeah, I mean, we think about that as token scarcity rather than imbalanced data, just to be clear. So in some sense it's inherent, and these generative pre-trained transformers are totally comfortable with variable frequency of tokens. There's nothing kind of weird about it. But that goes to the fact that we believe that you need enough instances, and empirically, it's somewhere between 500 to 1,000 instances of a disease token in your data set for us to make a good prediction. And then for you to assess that prediction, you need, hundreds, at least hundreds, if not multiple hundreds in your zero-shot validation data set for this. So that there is a kind of limit for a data-driven limit for this, but we think this will scale pretty linear. We think it's going to just scale. That everything we're doing is suggesting this will just scale. So if we put in 55 million people, we will be able to look at rarer events.
Thank you. Another question is, so this is an exciting approach to predicting disease. Does it have the capacity to identify factors that led to health? Adding, presumably, data related to healthy people are underrepresented in the training sets.
I mean, actually, it's the opposite. The bias in UK Biobank is for healthy people, richer, healthy, middle-class people. Both my parents are in UK Biobank. So we have this volunteer bias, which has been very, very often seen in cohorts. And actually, in the paper, I encourage you to look at the paper. We explore the biases quite thoroughly. Here is a very obvious bias, ascertainment bias, classic piece of epidemiology. UK Biobank was recruited between the ages of 40 and 60 as healthy individuals. And therefore, by definition, no one in the UK Biobank died before the age of 40. And unsurprisingly, our model refuses to let anybody die before the age of 40. I mean, it's considered to be a complete impossibility in the model's universe for anybody to die. That's obviously a bias and an incorrect aggregate prediction. But it's very much a data-driven case. But actually, yeah, if anything, the model's biased towards healthier people. And I encourage you to read the rather extensive section about ascertainment biases in the paper.
Yeah, thank you. Maybe I can get back to these cancers in Denmark. I mean, why units have been rather disappointing. I mean.
Gosh, we really, and the Finnish data has shown that we don't have a problem predicting cancer. I mean, some cancers are harder to predict, but we don't have an aggregate problem. There is a really complicated story here. Okay, just a reminder that the UK Biobank was recruited in an age window. It's 40 to 60. In fact, most people were recruited between the ages of like 47 and 55. It's quite a tight age window. We are basically at peak cancer time at the moment for those individuals. So their cancer incidence rates are going up. However, all systems have a delay between the disease happening and it being reported in the records. And there's some argument that that's even stronger for cancer, but you don't even have to make that argument. And the fact that we have this exponentially increasing cancer rate and then this delay means observationally the data goes up with age and then it has this artificial flattening. And we don't believe that's real. When we go forward a number of years, we believe that will go back up. But our model has correctly modeled that in age. Yeah. Now, the important thing to realize in Denmark is that it was a much broader age range. And so we don't have the same combination, this ascertainment, this very specific time-based ascertainment. So we think it's that simple. We really think it's that simple. I've just summarized 6 months of scratching heads and worrying about stuff. And, so we looked at pretty much everything else. And then we said, oh, my God. So it's a real classic piece of epidemiology at some level.
Thank you.
Thank you a lot. I don't have any more questions for now, so don't hesitate. And if not, we can maybe close this session here. Many thanks, Iwan, for joining us today and for your presentation. It was really interesting. I'll be happy to have you another time, maybe on another subject. So maybe I can wish you a good afternoon to all. Thank you a lot. Bye.